Imagine your body as a bustling city. Within this city, countless proteins act as workers, couriers, and builders. They carry messages, construct new cells, and defend against invaders. But sometimes a worker goes rogue. A protein might malfunction, causing a traffic jam in your cellular pathways. This is the root of many diseases. For centuries, our quest to find new medicines has been like searching for a single specific key for a single specific lock in a city with billions of them. It's a monumental task, one that has been painstakingly slow and incredibly expensive. Scientists have had to rely on a combination of educated guesswork, trial and error, and sheer luck to find a molecule that fits just right. The challenge lies in the complexity of these interactions. Proteins are not simple, static shapes. They are vast, three-dimensional structures that fold and flex in intricate ways. A potential drug molecule must not only fit into a specific pocket on a protein, but also bind to it with just the right amount of force to either block or activate its function. To find these perfect matches, researchers have traditionally had to synthesize thousands, sometimes millions of compounds, and test them one by one in the laboratory. This process can take years, even decades, and the vast majority of these potential keys will fail to turn the lock, leading to dead ends and wasted resources on the long road to a new cure. Think of it like trying to dock a spaceship with a spinning, tumbling space station blindfolded. You know the general shape of the docking port, but its exact orientation and movement are a mystery. You could send out millions of tiny probes, hoping one will randomly connect. This is the essence of traditional drug discovery. It is a brute force approach born of necessity, a testament to human persistence in the face of overwhelming odds. Every medicine on the shelf today is a survivor of this grueling journey, a tiny victory in a vast cosmic lottery. But what if we could take off the blindfold? What if we could see the docking port clearly? This is where the power of computation enters the story. For years, we have tried to simulate these molecular dances using powerful computers. These physics-based simulations try to calculate every tiny push and pull between atoms, a process of immense complexity. While incredibly detailed, these simulations are also incredibly slow. Predicting how a single drug molecule might bind to a single protein could take days or even weeks of supercomputer time. This bottleneck has limited our ability to explore the vast universe of possible medicines, leaving countless potential cures undiscovered, lost in the computational dark. We needed a new way to navigate this molecular cosmos. In the great expanse of scientific discovery, sometimes a new tool emerges that changes our perspective entirely. It's like the invention of the telescope, which allowed us to see beyond our terrestrial horizons and into the cosmos. In the microscopic universe of drug discovery, a new star has just appeared. It comes from the brilliant minds at the Massachusetts Institute of Technology, or MIT, and it's called Boltz-2. This is not a telescope for the stars, but for the very building blocks of life. It's an artificial intelligence model designed to do something extraordinary, to predict with incredible speed and precision how a potential drug will interact with a protein inside our bodies. Boltz-2 is a revolutionary step forward. It belongs to a class of AI known as a deep learning model. Instead of being explicitly programmed with the laws of physics, it learns them by observing vast amounts of data. Imagine showing a student millions of examples of successful key and lock pairings. Eventually, the student starts to understand the underlying principles of what makes a good fit, developing an intuition that goes beyond simple rules. Boltz-2 has been trained on a massive data set of known molecular interactions, allowing it to recognize the subtle patterns and geometric relationships that determine whether a drug and a protein will bind together successfully. The model doesn't just look at the static shapes of the molecules like a simple jigsaw puzzle piece. It understands that these are dynamic, three-dimensional objects. Boltz-2 uses a sophisticated technique that considers the geometry and rotation of both the drug molecule, the ligand, and the protein's binding site. It can essentially see the molecules in three-dimensional space, rotating and positioning them to find the most stable and energetically favorable connection. This is a crucial leap, as the way a molecule orients itself is fundamental to its biological effect. A key inserted upside down will not open a lock, no matter how well its shape matches. This new AI represents a fundamental shift in our approach. We are moving from a world of slow, methodical calculation to one of rapid, intelligent prediction. Boltz-2 is not just a faster calculator, it's a new kind of scientific partner. It can sift through immense digital libraries of potential drug compounds containing billions of molecules and flag the most promising candidates for further investigation. It acts as a guide, pointing our attention towards the most likely paths to success and helping us avoid the countless dead ends that have plagued drug discovery for so long. It is, in essence, a new map for the molecular universe. The real game-changing aspect of Boltz-2 is its incredible speed. To truly appreciate this, we must compare it to the old ways. Traditional physics-based simulations, the gold standard for many years, are powerful but painstakingly slow. They are like master craftsmen, meticulously carving a sculpture one chisel strike at a time. To simulate the binding of a single drug to a protein might take a supercomputer several days. Now imagine you have a library of 1 billion potential drug molecules you want to test against a single disease-causing protein. Using the old method, this task would be practically impossible, taking thousands of years of continuous computation. It was a barrier we simply could not cross. Enter Boltz-2. This AI model achieves a velocity that was previously the stuff of science fiction. Where the old methods took days, Boltz-2 can provide a prediction in mere seconds. It is more than 1,000,000 times faster. Let that sink in. What once took an entire weekend for a supercomputer can now be done in the time it takes to blink. That library of 1 billion molecules, Boltz-2 can screen the entire collection in a matter of days, not millennia. This is not just an incremental improvement, it is a quantum leap. It transforms drug discovery from a slow, deliberate crawl into a high-speed sprint. But speed is meaningless without accuracy. A fast prediction that is wrong is worse than useless. It's misleading. This is where Boltz-2 truly shines. Despite its blistering pace, its predictions are just as accurate, and in some cases more so than the much slower traditional physics-based methods. The team at MIT rigorously tested the AI against established benchmarks, comparing its predictions to real-world experimental results. The outcome was clear. Boltz-2 could identify promising drug candidates with a level of precision that rivaled the old guard. It has mastered the art of being both the hare and the tortoise, winning the race with both speed and reliability. This combination of speed and accuracy fundamentally changes the workflow for scientists. Researchers can now explore vast chemical spaces that were previously inaccessible. They can iterate on ideas faster, an essential part of the scientific process. If a potential drug molecule isn't quite right, they can ask the AI to screen millions of similar, slightly modified versions to find a better fit. This creates a rapid feedback loop, allowing for a more dynamic and creative approach to designing new medicines. It's like giving a composer the ability to hear an entire orchestra play their new symphony instantly, rather than having to teach each musician their part one by one. So, what does this breakthrough truly mean for all of us? The implications are profound, touching everyone from the scientist in the lab to the patient waiting for a cure. For researchers, Boltz-2 and similar AI models act as powerful accelerators. They can now pursue more ambitious projects, tackling diseases that were once considered undruggable because the target proteins were too complex or poorly understood. Scientists can test more daring hypotheses and explore unconventional molecular designs, knowing that the AI can rapidly sort the promising ideas from the dead ends. This frees up their time and resources to focus on the most critical next steps: laboratory testing and clinical trials. The most significant impact will be on the speed at which we can respond to new health crises. Consider the recent global pandemic. The race to find treatments and vaccines was a race against time. With tools like Boltz-2, the initial phase of identifying promising antiviral compounds could be compressed from months or years into just a few weeks. This could allow us to develop effective treatments much faster in the face of a new or mutating virus, potentially saving countless lives. It empowers us to be proactive rather than reactive, to get ahead of diseases before they spiral out of control. Ultimately, this acceleration in the early stages of drug discovery could lead to more affordable medicines for everyone. The journey of a drug from the laboratory to the pharmacy is incredibly long and expensive, with a high failure rate. These costs are inevitably passed on to patients. By making the initial discovery phase faster, cheaper, and more efficient, AI like Boltz-2 can help reduce the overall cost of developing new drugs. This could make life-saving treatments more accessible to people all over the world, regardless of their economic circumstances, helping to close the gap in global health equity. We stand at the dawn of a new era in medicine, one where artificial intelligence becomes an indispensable partner in our quest for health and longevity. Boltz-2 is a brilliant example of how human ingenuity, combined with the power of computation, can unlock secrets hidden within our own biology. It is a tool that allows us to navigate the molecular cosmos with newfound clarity and speed. As these AI models become more powerful and more widely used, the dream of rapidly finding cures for cancer, Alzheimer's, and countless other ailments moves ever closer to becoming a reality. The future of medicine is being written now, and it is being written in the language of code.